Evolution. Evolution, genetic change in a population of organisms over time. Evidences of evolution. 1. Fossils. 2. Comparative anatomy and structures. 3. Embryology. 4. Biochemistry, proteins and DNA. Evidence from fossil records. Fossils are traces of organisms that lived in the past and were preserved by natural processes. The preserved remains or traces of an organism that is no longer living. Usually found in sedimentary rocks. A. Carbon dating method used to determine the age of the rocks. Carbon-14 breaks down every 5,730 years. Types of fossils. 1. Imprint-thin, soft object, leaf, feather, is buried and sediments later harden. 2. Mold. Buried organism disappears and leaves an empty space. 3. Cast. Mold filled by minerals, replica of organism. 4. Petrification. Minerals replace hard parts, bones, teeth, etc., of organism. 5. Amber. Entire organism fossilized in tree sap. 6. Frozen. Entire organism frozen in ice. 7. Trace. Footprints, trails, etc. Determining a fossil's age. Relative age dating helps scientists determine the relative order in which species have appeared on Earth over time. To measure the age of sedimentary rock layers, scientists calculate the ages of igneous layers above and below them. If the age of the igneous layers is known, it is possible to estimate the age of the sedimentary layers and the fossils they contain between them. Geologic time scale is a chart that divides Earth's history into different time units. Era is the largest division of geologic time scale, namely Precambrian, Paleozoic, Mesozoic and Cenozoic. Each era is divided into period. The fossil record is evidence that horses descended from organisms for which only fossils exist today. 2. Evidence from comparative anatomy. Study of anatomical structures to find similarities and differences. A. Homologous structure, parts with similar basic structure, derived from same structures in embryo, same common descent, but may vary in function. The forelimbs of these species are different sizes, but their placement and structure suggest common ancestry. B. Analogous structure. Body parts that perform a similar function, may look somewhat alike, but differ in structure and different origin. Organisms evolve to the environment in similar ways. The comparison of analogous and homologous structures. C. Vestigial structures. Reduced body parts, in comparison to the same complex structure in other organisms, that have little to no function, remnant of an ancestor. Are body parts that have lost their original function through evolution. Examples: 1. Human appendix, other mammals it is necessary to aid in digestion. 2. Human external ear muscle, useless, but still there. 3. Human tailbone, coccyx. 4. Human wisdom teeth. 5. Bird wings. Penguins adapted for swimming, ostrich wings for balance and courtship. Vestigial structures of whales. Vestigial structures of cave fish and snake. 3. Evidence from embryonic development. Embryo. Early stage of development in organisms. Embryology. The science of the development of embryos from fertilization to birth. Closely related organisms exhibit similar embryonic development. All vertebrate embryos exhibit pharyngeal pouches at a certain stage of their development. These features, which develop into neck and face parts, suggest relatedness. Patterns of embryological development can indicate a common ancestry. Fish, birds, mammals and reptiles all have gills, only fish retain theirs. Fish, birds, humans and reptiles all have tails, all but humans retain theirs. 4. Biochemical analysis and amino acid sequence of the organism's DNA. The greater the similarity in amino acid sequence, the closer the relationship of the organisms. The organisms which are similar in structure and also possess similarity at the biochemical level could possibly have a common ancestor. Organisms with similar DNA share common ancestors and are closely related. Similarity of proteins, RNA and DNA molecules. Universality of genetic code, mRNA code. Supports evolution. Similar chemistry and structure of chromosomes among eukaryotes. Chlorophyll is the same basic molecule in all photosynthetic organisms. Protein analysis or amino acid sequencing. The numbers represent the number of differences between protein samples. Percentage close to human.